this functional property of pK1, pK2 and average you can calculate as a pK3, right? So pK1 and pK2 and then upon 2. So what will be the value? You can make them plus and then divided by 2. Okay. So now you can see the next one. This is side chain containing hydroxyl group. Only 3 are there. Serine contain hydroxyl group, threonine contain hydroxyl group, and tyrosine. Tyrosine is mentioned along with the aromatic side chain. So these two are aliphatic side chain that contain the hydroxyl group. CS2OH hydroxymethyl group is present in the serine, right? And in the threonine you can see CS3CHOH. CS3CHOH. So you have to remember like this. And also they have 2 pKa in which side you can see the this pK value, pK1 carboxyl side and pK2 amino group, and average pK3 is also existing about 30. Okay, some there are many amino acids which don't have the pK3. So if you will see the sulfur containing amino acid, this is a cysteine. This is very important because they contain the SH group and they are participating in many kind of amino acid. Okay, so polar uncharged group you can see methionine. Methionine is the non-polar. Methionine is the non-polar. CHSCS3. This is also containing side chain. Methyl, ethyl, thiol, ether, R group. And in the cysteine you can say thiol, methyl, sulfhydryl, R group. So these all side chain you must remember it well along with their compositions. They have a hydroxyl group or they contain benzene group, or the aliphatic side chain. Now here you can say side chain containing acidic group or their amide, aspartic acid. They have 2 carboxyl group as I was discussing yesterday. Okay, and if you see the asparagine, now you can also remember, you should also remember the SART form. Aspartic acid have a SART name as a D. Okay, you can see here the SART name, asparagine N. Okay, glutamic acid as E, glutamine as Q. So you must remember it, right? And asparagine side chain is this one, CH. This attached to this branch group CH2CO and S2, the asparagine. And here also you can see there is a 1 carboxyl, 1 amine, and other extra group is CO. Okay, so that is the composition. Why they have the pK3? Because you can see there are 2 carboxyl and 1 amino acid. Okay, so wherever there will be 2 carboxyl acid, you can see there will be 3 pK, and pK1, pK2 as well as pK3. This belongs to amino acid and this will belongs to carboxyl group. Okay, and you can say the glutamine Q, they are classified under the basic amino acid, means they have 2 NH2 group. Okay, 2 NH2 group and 1 carboxyl group. Okay, but still they have only 2 pK, pK1 and pK2, right? So what is the short form of arginine? R. Abbreviated form of lysine? K. Abbreviated form of histidine? H. Okay, so now you can see what is the composition of arginine. What is the composition of arginine? Tell me. There are how many NH3? Just wait. Arginine contains how many NH group? NH3 as well as NH2 plus. Okay, so this special group is called as guanidinium R group. What is the name? Guanidinium R group. Okay. So arginine, guanidinium R group. Is it clear? Any doubt is there? You can ask me. Okay. Lysine. How many NH2 group? NH3 group? There are 2 NH3 group. But since alpha, beta, gamma and beta. Okay. So this is a beta NH3 group attached to the lysine. That's why this is very special. Arginine is special because they are containing a special functional group that is called as guanidinium R group. Clear? And he studied why this is special because they contain imidazolium. Many drugs have found to contain this one. Okay. So they are physiologically very relevant when you are studying their pK value, and also they are imparting special features to the proteins, various kind of proteins. So now coming to the aromatic amino acid containing the aromatic ring, for example histidine H, phenylalanine F. मेरे साथ साथ बोल सकते हो, no problem, but that will help you in learning, and also you can note it down. Okay. So phenylalanine F, benzene ring you can see, tyrosine Y, phenol R group is there. Okay. Tryptophan, heterocyclic structure, endole R group is there. That's a very specific. Tryptophan written abbreviated as W, tyrosine abbreviated as Y. Okay. Clear?
Yes, sir. It is a neuro.
Any doubt?
Side chain molecule.
And amino acid proline. Proline contains proline, specific amino group that belongs to 5-member array. So these are some special features. And what are the functions of essential amino acid? You can see. For example, phenylalanine. These are the precursor of tyrosine. Right? Phenylalanine is the precursor of tyrosine. Tyrosine, dopamine, you know. What is dopamine? Neurotransmitters present in the brain. Okay. Dopamine, norepinephrine, this is hormone, epinephrine, melanin, all these are made up of phenylalanine, right? Phenylalanine. And what is the genetic disorder caused? Phenylketonuria. Phenylketonuria is the inability to metabolize phenylalanine because of lack of enzyme. Many times this has been asked in the CSIR examination. Okay? And many, not only CSIR, DVP, ICMR, every examination they are going to ask phenylalanine hydroxylase. What is their function? You know? So because phenylalanine hydroxylase is lacking, that's why they cannot utilize phenylalanine amino acid. Because of this essential amino acid, there is a genetic disorder called as the phenylketonuria, right? And if you are talking about the tryptophan, then tryptophan is also essential amino acid, right? And what is the role? Have you heard about that? You heard about that? Tryptophan is right, yes. The tryptophan precursor for neurotransmitter, serotonin, very important, hormone as melatonin, and various vitamins like niacin, tryptophan, tyrosine DHT anchoring membrane protein within cell membrane. So phenylalanine as well as tryptophan precursor of neurotransmitter. Many times this has been asked. Serotonin is found by which precursor? So this is tryptophan. Melatonin found by which precursor? Tryptophan. Vitamin niacin is found by which precursor? Tryptophan. So if tryptophan is lacking, then many times you can develop which kind of disease. You can see neurotransmitter will be lacking. Okay. This hormone will be lacking. Melatonin gives the dark color to the body. Okay. So many people you can see they are without melatonin. So what is the effect? Their composition is very white, right? So they cannot sustain, they cannot protect themselves from the UV rays, harmful effect of the UV rays. Okay. So fructose metabolism causes fructose malabsorption causes improper absorption of tryptophan in intestine causes reduced level of TRP in blood. Okay. So fructose malabsorption causes improper absorption of tryptophan in intestine, and that causes reduced level of tryptophan. So these both are linked, fructose as well as tryptophan. Okay. So in aliphatic amino acid, alanine synthesized from pyruvate. Pyruvate, the part of metabolism of carbohydrate. Okay. And also alanine is the precursor of branched chain amino acid. Okay. And it plays a very important role in glucose-alanine cycle. Many times this cycle is asked. What is glucose-alanine cycle between tissue and liver? Okay. So then we will discuss when we are covering the metabolism part. Okay. Yeah. So this cycle enables pyruvate and glutamate to be removed from muscle and safely transported to liver. This is important. Now you are, I think, clear about this role of cycle, glucose-alanine cycle. What is the role of glucose-alanine cycle? This cycle enables pyruvate and glutamate to be removed from muscle and safely transported to liver for further metabolism. Okay. And when there is an alteration in the alanine cycle, then there is a problem. Then there is a high level of ALT. ALT means alanine transferase, right? And if alanine transferase is high, it is going to develop a disease called as the type 2 diabetes. Nowadays, maximum people are developing this kind of disease, type 2 diabetes, because of alteration in alanine cycle and increased level of high level of what? High level of ALT. Got the point? Is it clear or not? Tell me. Is this clear, better? Hello. Okay. ठीक है. चलो. Valine essential for hematopoietic stem cells, self-renewal in cell cycle disease. You know already how cell cycle disease occurs because of replacement of some amino acid. Here glutamic acid in beta globin are replaced with valine because valine is hydrophobic. Therefore, glutamic acid is also hydrophilic. This change makes the hemoglobin prone to abnormal aggregation. So that's why valine is essential for HSC self-renewal. What is HSC? Hematopoietic stem cell. Means hematopoietic cells are going to be formed. And also valine may be the cause of sickle cell disease. What is the symptom of sickle cell disease? Hemoglobin. They are prone to abnormal aggregation, right? So leucine primary metabolic end products of leucine metabolism are acetyl coenzyme A and acetoacetate. It is a very important ketogenic amino acid. What is ketogenic amino acid? This was asked in the last CSIR exam.
Yes, sir.
What is a ketogenic amino acid? So leucine is one of the ketogenic amino acids. Ketones are formed because of primary end product metabolism, right? So when there is a primary end product metabolism, will be product will be acetyl coenzyme and acetoacetate. This will be called as the ketogenic amino acid. Is it clear, beta? So valine, you can see they are having the role in hematopoietic stem cell self-renewal. This is very important also because nowadays this is the current hot topic, hematopoietic stem cell. Okay, and obviously CSIR people are targeting those questions which are related to the metabolism discussion. Mostly these questions are there. Okay, for example, what is sickle cell disease? What is hematopoietic stem cell? How they are going to be developed? Which cytokines are involved? Which hormones are involved? A lot of questions are going to ask. That's why I thought I should not leave this topic. Okay, so now you can see leucine. This is also ketogenic amino acid I have discussed just now. And why this is ketogenic amino acid? Because they are forming acetyl coenzyme A and acetoacetate as a end product. Okay, so adipose and muscle tissue are using the lysine for the formation of sterols. लोग कहते हैं ना काफी मोटे हो जा रहे हैं, तो मोटे होने का रीजन क्या है? That is ketogenic amino acid. Ketogenic amino acid अगर हाई होगा तो there will be lot of crystal coenzyme and acetate, adipose and muscle tissue uses this leucine, ketogenic amino acid as a sterol formation. That is a good fat or bad fat? Can you tell me sterols?
Bad. Because these are steroids.
This is good fat or bad? My voice is breaking. Now it's clear? Is it bad or good? I'm asking you.
Bad.
Yes. It depends on the composition. If this is a fufa, then it's a good sterol. If there is so many linear chain, then it's kind of bad cholesterol, bad sterols. Okay, cholesterol. So toxic product can be formed if there is a high amount of keto acid present in the blood and urine, and if your these amino acid intake is very high, then obviously they are going to be release a lot of toxic product, right, in the blood and urine, and that is very harmful for our metabolism, our body, and our systems, right? So just a minute. Isolation, they have the diverse physiological function, for example in blood clotting and stimulating immune function, promoting secretion of several hormones. So that's why they are called as the essential amino acid. Sulfur containing amino acid, already substrate for other amino acid like cysteine, taurine, and they are very versatile for SAM. What is the SAM? Have you heard about this amino acid compound? The SAM, if you will see in the molecular biology, so what is happening there? As soon as the new DNA is going to form or new protein is going to form, they are immediately methylated by the SAM. Okay. So this is used for the purpose of methylation of newly formed DNA, so that they should be protected. Otherwise, there are internal enzymes which are going to believe them as a foreign DNA, and immediately they are.
This SAM, is it
That's why
useful in
there should
splicing
be.
or methylation of DNA? Something.
This is acting as a marker of the DNA. This is because of methionine.
Achha.
Lysine, what you are asking about lysine?
So I'm asking that the SAM, is it useful in splicing the methylation of DNA?
Slicing में नहीं बेटा, ये मार्कर होती है कि ये अपना डीएनए है. This is used for distinction of our own DNA to foreign DNA. So if this is not methylated, if any DNA is not methylated, it is assumed as it is a foreign. तो अगर foreign है तो उसको slice करेगा. Got the idea? So now homocysteine can be regenerated from methionine itself, or they can be used to form cysteine. So if there is improper conversion of methionine, that can lead to arthrosclerosis, very common disease, arthrosclerosis, and this is because of accumulation of homocysteine. This is because of homocysteine. Clear? Polar uncharged amino acid. Threonine, its residue susceptible to numerous post-translational modifications. Okay, threonine susceptible to numerous post-translational modifications. Modification is very important for it. And every year you will see, every time they are asking about this CSIR, DVTS, but every examination they are asking. What is the O-linked glycosylation? O-linked glycosylation means there is a additional hydroxyl side chain. Okay, that is undergoing the modification, and they are going to form O-linked glycosylation. Okay, so threonine residue under go phosphorylation through the action of threonine kinase. है ना? Phosphorylation is very important activity in the modification of various proteins, various kind of function modification of. So in its phosphorylated form, it can be preferred as phosphothreonine. Its role in cell signaling. Okay, it's having a vital role in cell signaling, neural activity. Okay. What is the polar charge amino acid? Positive charge basic amino acid, histidine, precursor of histamine, and amine produced in the body necessary for inflammation. Histidine, ammonia ligase convert histidine into ammonia and uracanic acid. And this deficiency in this enzyme is a kind of rare metabolic disorder called as histidininemia. Histidininemia, right? I think you are hearing this kind of disease first time, no? So this is all advanced biochemistry, no? We are covering for the CSIR. From last 3 years, I am teaching online mode because offline has a various barriers. So this is very tough for a teacher to survive in the offline mode. The student number is not very good. Okay, online mode you can survive. You can go through with the 1 student, 5 student, 10 student. Lysine, another amino acid, I have discussed that they have the beta amino group. So their contribution is asked in the CSIR examination. They are contributing as participation in hydrogen bonds, salt bridges, and covalent interaction to form CIF base. Right. So can you remember this? Lysine. I'm very fast
it will
or
be
slow.
Okay. थोड़ा सा slow
Or its speed is correct.
I'm comfortable.
Okay.
It's okay. Okay. So a little bit you need slower or this much is.
Yeah, sir. It will be okay.
Okay.
थोड़ा सा slow.
चलो ठीक है. तो
I'm comfortable.
हाँ, लाइसिन में देख सकते हो ऑलरेडी वी हैव डिस्कस्ड बीटा अमीनो ग्रुप. सो ये बड़ा इंपॉर्टेंट है. दे आर फॉर्मिंग कंट्रीब्यूटिंग इन साल्ट ब्रिजेस फॉर्म. जब भी लाइसिन आ जाए, लाइसिन से रिलेटेड कोई प्रोडक्ट आ जाए तो इमीडिएटली दे आर आस्किंग व्हाट काइंड ऑफ कंट्रीब्यूशन आर देयर. तो आपको देखो कि तीनों कंट्रीब्यूशन दे रहे हैं, राइट, नॉट सिंगल. हाइड्रोजन बॉन्डिंग एज वेल, साल्ट ब्रिजेस सेकेंड, एंड थर्ड विल बी द कोवेलेंट इंटरेक्शन. ठीक है ना? I will be sharing the video recording properly. I don't know. I'm using the two-three recorders. I don't know what will be recorded because I am using the first time as a presenter. As a listener, I have participated in more than 100 webinars, but first time as a presenter I am using this. So I think video will be very good. No problem. So second major role of lysine, you can see epigenetic modification. Have you heard about the epigenetic modification?
No sir.
So you are from zoology background or botany?
I'm from zoology background only, but I haven't heard this song.
You are in the third semester, na?
No sir, I'm in master's first semester.
Okay. So epigenetic modification, this is the current perspective, very hot topic, because aging in the person. Aging is person, you can stop the aging, the person will be forever young. And if you are going to change this, epigenetic regulation, which is by means of histone modification. There is a histone modification by adding the methyl group. Okay, so then whenever the methyl group is added, this is called as the epigenetic modification. So there may be two processes, removal of methyl group and addition of methyl. Both are part of epigenetic modification. So except India, the whole world is studying over this. India is studying over because facility is very less.
जैसे टेलोमेरेज एक्टिविटी होती है, वो भी हम कहते हैं कि एजिंग के साथ टेलोमेरेज कम हो जाती है.
वो तो देखो होल ईयर, मतलब जब आप बड़े होते हो तो एवरी ईयर, एवरी टाइम यू कैन से टीलोमेरेज इज करेक्टिंग द लेंथ ऑफ द टीलोमियर्स. जब टीलोमियर्स ये देखा गया कि जिस सेल में शार्ट हो जाती है, जिस एनिमल में शार्ट हो जाती है, उनकी लाइफ घट जाती है. तो वो लाइफ बढ़ाने से रिलेटेड है, ठीक है ना. दैट इज नॉट द पार्ट ऑफ एजिंग। आपकी लाइफ बढ़ती है, जैसे 70 साल जीना है, 50 साल जीना है ना, वो वाला है। इपिजेनेटिक मॉडिफिकेशन जो आपके स्किन का वो टोन कैसा होगा एट द एज ऑफ 70 ईयर्स। आपने देखा होगा बहुत सारे लोग होते हैं, जापानीज लोगों को अक्सर देखो आप, है ना, जापानीज आर द बेस्ट इन द वर्ल्ड इन द टर्म ऑफ देयर फिजियोलॉजिकल कंट्रोल। उनकी, हमारे यहाँ इंडिया की जो मैक्सिमम एज, लिविंग एज होती है वो होती है 60 to 70। आपने देखा होगा ज्यादातर फिल्म स्टार तो 35 to 50 में खत्म हो जा रहा है ना। उनकी एज तो काफी कम है। हमारी जो जनरल पब्लिक है वो चूंकि बहुत हार्मफुल चीज यूज नहीं करते हैं, बहुत सिंथेटिक चीज यूज नहीं करते, तो दे आर, तो उनकी एवरेज लाइफ एक्सपेक्टेंसी होती है 70 ईयर्स। ठीक है ना।
कोलेजन का हो सकता है एपिजेनेटिक रेगुलेशन?
नहीं, वो हमें पता है कि आप किस वे में बात कर रहे हो। फॉर एग्जाम्पल बहुत सारे कंपनीज वो सेल करते हैं ना, कोलेजन लगाओ बॉडी में, है ना, तो वो स्किन का टोन चेंज कर देगा। हाँ, आप एज ए लाइफ साइंस स्टूडेंट सोचते हो ना कि कोलेजन हम बाहर से लगा के हम क्या कर लेंगे, अगर इट्स नॉट ए पार्ट ऑफ इंटरनल सिस्टम, है ना, तो वो तो बेवकूफ बना के कंपनियां बेचती है। जैसे उनको कोई बायोलॉजी, जैसे हम लोगों जैसा कोई ज्वाइन किया होगा रिसर्चर, तो उनको बताया होगा कि यार कोलेजन का बड़ा इंपॉर्टेंट रोल है ना। तो वो जब बता रहा था तो उसने वो बात करी कि भाई इंटरनली दिस इज गोइंग टू प्ले वेरी इंपॉर्टेंट रोल, है ना। उनको समझ में आया बाहर से बना के बेचो। तो बाहर से बना के हाउ यू कैन डू दैट, दिस इज टोटली इम्पॉसिबल। ठीक है ना। बाहर से कोलेजन लगा के, जैसे मान लो देखो, मैं ये कहता हूँ कि अब एजिंग पे बात तुमने करी है तो मैं अपना एक्सपीरियंस शेयर कर देता हूँ। व्हाट इज स्किन टोन? स्किन टोन मतलब अगर बहुत ज्यादा हम धूप में दौड़ रहे हैं, बहुत ज्यादा पॉल्यूटेड एरिया में है, तो ऑब्वियसली जो हमारा मेलाटोनिन है वो डार्क कलर देगा, है ना। अभी इंडिया के लोगों को आपने देखा होगा, इफ दे आर माइग्रेटिंग टू यूके, यूएस, ऑस्ट्रेलिया, न्यूजीलैंड, उनका फेयर स्किन हो जाता है आफ्टर 4 to 5 ईयर्स, क्योंकि मेलानिन बनना बंद हो जाती है आपको, है ना। तो उनकी स्किन उन्होंने कोई चेंज नहीं कराया, बट जो इन्वायरमेंटल कंडीशन है दैट इज अफेक्टिंग योर मेटाबॉलिज्म। ठीक है। तो अगर इपिजेनेटिक मॉडिफिकेशन चाहते हो तो जैसे जो अरहर की दाल सुना है आपने,
में भी होता है,
अरहर की दाल में होता है ये लस, बहुत ज्यादा होता है। ठीक है ना। तो अगर आप ये लेते हो, नहीं, ये मैं नहीं, ये दालों में होता है, पल्सेस में होता है सिर्फ, और ये अरहर की दाल में होता है। अरहर की दाल देखो बहुत सारे स्टेट में नहीं खाते हैं लोग, तो उनकी प्रॉब्लम होती है इपिजेनेटिक मॉडिफिकेशन। अगर आप खाते हो तो आपका पॉजिटिव वे में मॉडिफिकेशन, योर एज माइट बी मोर देन हु आर नॉट ईटिंग दीज। ठीक है। और आपका स्किन टोन, कलर, अगर देखो बहुत सारी नेचुरल चीजों को खाते हो आप, विटामिन, अमीनो एसिड्स सारे ले रहे हो, तो देयर विल बी नो प्रॉब्लम, है ना। तो प्रॉब्लम है हमारे लाइफ स्टाइल की, ईटिंग की, हम खाते क्या। एंड वी शुड नॉट बी अफेक्टेड बाय द एडवर्टीजमेंट, बिकॉज 99% एडवर्टीजमेंट आर फॉल्स, दे आर क्लेमिंग फॉल्स, है ना, दे आर टेलिंग फॉल्स, there is no scientific evidence, they are not giving you any data, and if you are searching over the internet also you will not going to find any truth among that, है ना। जैसे तुम्हें बताऊं, एक सेंसोडाइन कंपनी थी दंत मंजन की, है ना, तो दैट कंपनी वाज सेलिंग कि जब झंझनाहट होती है दांतों में तो आप मेरा मंजन करोगे तो दूर हो जाएगी। अब झंझनाहट क्यों आती है, उसका रीजन अलग है ना। अगर आपको झंझनाहट आ रही है इसका मतलब कैविटी है आपके दांतों, तो जब भी आप खाओगे तो वो कैविटी में जाके फंस जाती है, तो वो जो वहां पे न्यूरॉन्स हैं दे आर वेरी सेंसिटिव, है ना, तो वो कह लो कि एक तरह से डिजनरेट होने स्टार्ट करती है, तो वो झंझनाहट आती है। तो इसका इलाज ये है कि कैविटी में फिलिंग करवा लो, है ना कि मंजन कर लो। तो दैट वाज द फॉल्स इंफॉर्मेशन। Information was correct but arranged in wrong way, है ना, arranged in wrong way। जैसे वो भी ट्रू था कि झंझनाहट होती है दांतों में, बट वो मंजन लगाने से दूर होगी इसका कोई वो नहीं था, प्रूफ भी नहीं था और कोई लिंक भी नहीं था कि
अभी
वो कैसे
100%
करेगा। कैविटी फिलिंग का रिप्लेसमेंट तो है ही वो।
उस टेस्ट का भी 100% टेस्ट
हाँ, इसीलिए उसको 5 करोड़ का फाइन लगा कंपनी। जैसे हमारे जैसे किसी साइंटिस्ट ने डाल दिया था कंज्यूमर फोरम, भाई ये 100% टेस्टेड लिखते हैं तो बताएं कहाँ पे है।
100% टेस्टेड।
तो 5 करोड़ कंपनी को फाइन लगा और 2 करोड़ उस बंदे को मिला जिसने ये डाला था कंज्यूमर फोरम में। अभी तुमने देखा होगा एक जैसे हाल ही में बहुत सारे शैम्पू पे बैन लगा है एंड ऑल दे वेयर कंटेनिंग द बेंजोइल ग्रुप। डू यू नो द फंक्शन ऑफ बेंजोइल ग्रुप? तो आजकल एक शैम्पू का ऐड आया होगा, बहुत पॉपुलर कंपनी है, आपने देखा होगा, वो कहती है कि भाई ड्राई स्किन के लिए, है ना, ड्राई स्किन के लिए। ड्राई स्किन के लिए तुम क्या दे रहे हो? बेंजील दे रहे हो। बेंजील कार्सिनोजेनिक है। बेंजील अगर लगाएगा बंदा तो उसको तो कैंसर ही हो जाएगा, वो तो मर ही जाएगा। और वो भी आप हेयर में कह रहे हो लगाओ, हेयर के थ्रू पूरे बॉडी में वो बेंजील टच में आ रहा है और उसकी कंसंट्रेशन भी ऐसा नहीं कि लो हो, है ना, बहुत हाई था। तो हमारे इंडिया में क्या है कि जब पूरे वर्ल्ड में कोई चीज बैन हो जाती है ना, तो सारी यूरोपियन कंट्री इधर ही आके अपना बिजनेस है, मतलब दे आर नॉट गोइंग टू क्लोज दैट बिजनेस। सोचते हैं इंडिया वालों में बेचो, ये बड़े अंधविश्वासी होते हैं बेचारे, जो कहो इनको मान लेते हैं, है ना, ठीक है ना। तो नाउ यू कैन सी रोल ऑफ लाइसिन। लाइसिन इज वेरी वेरी इंपॉर्टेंट करेंसी, मतलब आपकी आधा मेटाबॉलिज्म तो यही कंट्रोल कर रहा है। यानी जैसे हफ्ते में अभी मेरे को डायबिटीज है, ठीक है ना, तो आई एम नॉट गोइंग टू टेक टू मच राइस और कार्बोहाइड्रेट्स, लेकिन मैं दाल जरूर लेता हूँ, अपनी सब्जी बनाऊंगा, दाल भी बनाता हूँ, रोटी बनाऊंगा, और आई एम प्रिपेयरिंग ऑल दैट कीपिंग इन माई रिक्वायरमेंट्स। ठीक है, तो आपको भी खाना बनाना आना चाहिए। मेरे ख्याल से आज की डेट में मैक्सिमम 80% गर्ल्स को तो खाना बनाना नहीं आता है। एंड दिस इज फैक्ट। और जो खाना बनाना नहीं आता बिकॉज दे डोंट नो द वाइटल रोल ऑफ दीज थिंग्स। जो भी खाना बनाने वाली मेड होगी या जो कोई पर्सन होगा, तो दे विल कीप इन माइंड ओनली द टेस्ट। नूडल जैसे खाना खाते हो या पिज्जा खाते हो, उसमें एक कार्सिनोजेनिक केमिकल डालते हैं। आपको आइडिया है कौन सा केमिकल डालते हैं?
नो सर।
तो वो नहीं खाना, कोशिश करो कि जितना कम से कम खा सको, बिकॉज कम